NOD2 mutations and its association with Crohn's disease. Crohn's disease is a chronic inflammatory disease of the gastrointestinal tract. Along with ulcerative colitis, it is one of the two major forms of inflammatory bowel disease, or IBD. It can lead to abdominal pain, severe diarrhea, fatigue, weight loss, and malnutrition. Crohn's disease can be both painful and debilitating. There is no known cure, but with treatment, many people are able to function well. It wasn't until the 1930s that epidemiological observations suggested there was an inherited component to Crohn's disease risk. Family and twin studies that followed showed that approximately 15% of patients with IBD have first-degree relatives that also have IBD. In 2001, two studies, one by Huguet et al. And another by Agurda et al., localized the specific causal mutations to 3 coding variants within the NOD2 gene, which is located on chromosome 16. Huguet et al. Genotyped 25 Caucasian families with at least 2 affected siblings using polymorphic markers, and Agurda et al. Used case control analysis with 416 patients with Crohn's disease and 287 controls, and PCR analysis to further characterize familial transmission of the gene. Both studies found what is now one of the most notable mutations on the NOD2 gene, which is a C insertion mutation in exon 11 in the coding region. This insertion leads to a premature stop codon, and this truncation is thought to lead to a dysfunctional leucine-rich repeat domain. NOD2 is a member of a gene family which is active in immune cells that line the intestine and protect the body against foreign invaders through activation of the protein nuclear factor kappa B, which has a role in initiating inflammatory responses. The C insertion mutation has been shown to exhibit a 5-fold greater activation of nuclear factor kappa B than the wild type. Thus, the overproduction of the nuclear factor leads to chronic inflammation as seen in many patients with Crohn's disease. This mutation in NOD2 is the start to understanding some manifestations of Crohn's disease. While being homozygous does confer a high degree of risk, this mutation is also rare. According to a study by Hamp et al., they estimate about 18% of the genetic risk of acquiring Crohn's disease in the population can be attributed to this C insertion mutation in NOD2. So far, it seems that the NOD2 mutations might be European-specific, as there is not the same association found in genome-wide association studies of Chinese patients with Crohn's disease. Additionally, there might be environmental factors that contribute to these differences. Furthermore, geneticists have discovered over 200 genetic variants that influence Crohn's disease risk. Overall, the clinical implications of NOD2 mutations are in need of greater investigation, but this finding has led to some important treatments that target the nuclear factor kappa B pathways to reduce inflammation and thus help patients control their Crohn's disease symptoms to live healthy, pain-free lives.